Hey guys, this is me Akash here for Biology Vibe. So today we'll start with Vibe 1.8, that's translation. So now till transcription, the mRNA has been formed in the nucleus and it has moved out of the nucleus through the nuclear pore and it reaches our protein factory, that is the ribosomes. And now it has to be decoded. This decoding stuff is known as translation. The translation involves decoding of the messenger RNA and using its information to build a polypeptide chain or chain of amino acids. So this is an mRNA, this is your protein factory, that is ribosomes made up of two subunits, the larger one and the smaller one. This is tRNA that has the responsibility to carry out that specific amino acid to that messenger RNA. And then there's a synthesis of a polypeptide chain. Let's put down deep into that. Now, before that, let's review the structure of tRNA. The main important things to be kept in mind here are first, this region. This is your mRNA that has codons, right? Complementary to that, there is specific anticodon, and corresponding to this anticodon, there's a specific type of amino acid placed here. So there are 20 types of anticodons corresponding to that specific codons, and then there's a specific 20 types of amino acids. So you see 20 types of tRNA having different anticodons and have different amino acids. So this was about tRNA, a little bit review. So now the whole process starts with the charging of tRNA. Firstly, an important enzyme known as amino acid tRNA synthase comes into picture. It carries up a specific type of amino acid, this blue portion, a specific type of amino acids, and then binds to ATP. Okay. Now what happens? Pyrophosphate is being released, PPI. Two P's will be released, one P is still there, right? Now what happens? A specific type of tRNA that will take up this specific type of amino acid comes into picture, and AMP is released. If you see PPI is already released, what the thing is left with? Pentose sugar, adenine base, and one just PI. That is AMP. So AMP is also being released, and finally a charged tRNA, that is a tRNA with carrying that specific amino acid, is released. So this was about charging of tRNA. Now what happens is this, the protein machinery, that is the ribosomes, have to be there. So the smaller subunit binds to the mRNA at a specific site. There are some sequences that will recognize a specific site, and there's a start sequence. Remember AUG, which corresponds to an initiated tRNA that will carry methionine. So the translation process starts here in the initiation phase. A specific type of codon known as the start codon, that is AUG, corresponding to that there's a specific type of amino acid which will be carried by a specific type of tRNA which has anticodon UAC will come into picture, and the first amino acid is methionine here. So if you see, there's a larger subunit in the ribosome and there's a smaller subunit in the ribosomes. In the middle we have the mRNA. There are three sites in the ribosomes: E site, P site and A site. E site exit 1, P is peptide site, and A is amino acid site. So the first tRNA will come into picture and it will bind here, carrying a amino acid methionine here at the P site. Now the second amino acid will come to the A site. Now let's see how it goes. Second amino acid comes at A site, and what happens is the this amino acid binds to this amino acid, forms a peptide bond here, Remember, amino acids are joined together via peptide bond. So a peptide bond is formed between one amino acid that is present at the P site and the amino acid that's present in the A site. Okay, so a bond is formed between them, that is peptide bond, and the whole machinery slides over that mRNA and the process goes on. Now next thing, the transfer tRNA which was at the A moves to P, and a new tRNA comes into picture and binds with A. At the same time, one tRNA will be released. Now this tRNA which is being released does not contain any amino acid here, because the amino acid it has already been placed here. So this way again the whole cycle will elongation cycle will continue. So you see, this is the P site, moves on. Now at the A site, one tRNA will come, one charged tRNA will come, that will carry a specific type of amino acid corresponding to this codon. There's an anti-codon here, specific type of amino acid comes into picture, moves on, a peptide bond is being formed here, the machinery moves on. Now one tRNA, that is uncharged amino acid, uncharged tRNA will move out from the exit side, and the process will continue. This is known as elongation. Now the thing which comes into picture is the termination part. At the termination part, we have the stop codon, namely UAG, UAA and UGA. Three stop codons are there. For these stop codons, no specific tRNA is there, no specific amino acid is there, so no tRNA will come into picture and the whole machinery will be opened up, like the smaller subunit and the larger subunit and the mRNA and the free polypeptide chain will be released. Okay, and then all those folding and specific signaling molecules occur there, and it goes to specific parts. That's a different story. But till here, the translation machinery has been finished. This translation process has been ended. So this kind of gives you a whole overview picture. The transcription occurs, mRNA being formed, moves out of the nuclear pore, goes to the ribosomes. Ribosomes have three sites: A site, P site and E site. Starts with the P site, methionine AUG code start codon. Then the charged tRNA comes to the A site, a peptide bond is being formed and the machinery moves. One amino acid, that is the uncharged tRNA, will move out from the exit side, and at the same time one charged tRNA will come into picture and the process goes on. Finally we reach to the termination, where we get a termination codon, stop codon, for which no tRNA is there, so the process stops and the free polypeptide chain is being released. So that's kind of it, guys, for this lecture, and I hope you enjoyed this video. So if you like this video, please hit the like button, subscribe to my channel to get more and more videos like that. Thank you.